Our next topic is tRNAs. What are tRNAs used for? Well, they bring amino acids to the ribosome during translation and match them up with the codons of the mRNA. tRNAs are about 75 to 90 nucleotides long, and they form this cloverleaf structure, which you can see here. The 3' end is called the aminoacyl end since it's where the amino acids bond, and it ends in the sequence CCA. You can think of this as standing for can carry amino acids, since this is where the amino acids attach. The opposite end contains the anticodon, which is complementary to a codon in the mRNA. It's this anticodon-codon matchup that allows each codon to correspond to an amino acid. Before it can do its part in translation, a tRNA must first be charged by adding its corresponding amino acid to it. This is an ATP-dependent process catalyzed by an aminoacyl tRNA synthase enzyme, and there's a different enzyme for each tRNA. So here we have a tRNA that corresponds to methionine, because the methionine codon is AUG. So this might end up at the start codon, and the aminoacyl tRNA synthase for methionine will first check if this is indeed the methionine tRNA, and if it is, it'll attach a methionine amino acid to it at the 3' end. This step requires 1 ATP. Once the tRNA is charged with its amino acid, a protein called initiation factor 2 is needed to get it into the ribosome so it can find its codon. Since this is such a vital process, we can use antibiotics such as tetracyclines to target the 30S subunit on a bacterial ribosome and prevent attachment of the aminoacyl tRNA, and this will kill bacteria. There are a couple other parts of the tRNA anatomy that are less important, but they might show up in your Step 1 exam. For example, the T-arm contains a modified type of nucleotide called pseudouridine, which is represented by the Greek letter psi, and the D-loop has a modified nucleotide called dihydrouridine, represented by a D here and here. You might get a question on the Step 1 that basically just asks you what type of molecule might contain pseudouridine or dihydrouridine, and your answer choices will be something like DNA, mRNA, tRNA, microRNA, etc. And you just have to know that it's tRNAs that have these weird types of nucleotides. The other type of question you could get is about the function of these different regions, and everything you have to know about that is written out pretty clearly in your book. The T-arm is involved in attaching to the ribosome, and the D-loop is important for binding to aminoacyl transferase. I talk about tRNA wobble more in a different slide, but I'll briefly go through it again here. Basically, the idea is that there are only 20 amino acids, but there are 64 possible ways to combine 4 different nucleotides in a sequence of 3. So each amino acid tends to correspond to multiple different codons. For example, proline is coded for by all of these codons, which differ only in their third base. So if there's a mutation in that third base, you'll still end up with the same amino acid in the protein. Therefore, this third base doesn't matter as much as the other two. Now this isn't always the case. There are also some amino acids that are only encoded by one codon, but by and large, the ones that are encoded by multiple codons only vary in the third position.